on that she can get it. Right, we start first lesson mammalian gametes. Now, the last time we learned about the plant fertilization. So here we're going to learn about generally the fertilization process of mammals, right? That vertebrates. So first we're going to look mammalian gametes, a female gamete cell ovum. Okay, this represents the part of the female reproductive system, and you can see here the ovary is the place where all these ovums are produced, or ovums stored here. And then the ovary is not physically connected to oviduct, but there is a fibrous attachment, the ovary to the oviduct or fallopian tube. And you can also see there is a projections, large number of projections which can move around this ovary to collect the ovum. So if the ovum is released, this can be collected by these projections, and once it enters to the oviduct, the ovum can't, it's not a motile, it cannot move, but there is a large number of cilia inside the internal side of the oviduct, and also contraction of the oviduct, also the movement of cilia. This ovum is moving towards to the uterus. Right now, when this the ovum is released, we're going to see how this structure looks like, right? Okay, it is a cell, a round shaped cell, and this cell still undergoing a meiosis, because when there's a difference with the male and female. When the female is born, they have a fixed number of egg cells or ovum, maybe around 800,000, and they are already in the stage of a meiosis. That means these fixed number of cells started meiosis during the embryo period, but after that the meiosis process stopped, and it's not happening. Then after the puberty, then egg cells start to mature. So we are not focused on that side, the production of ovum. It is called oogenesis, but we are not learning that part here. But we're going to learn the cell and the fertilization. So when the ovum start to mature, so it starts from every month, and then it starts to complete the rest of the stage of meiosis. So mostly meiosis 1 already happened, and then meiosis 2 start. During the process of meiosis 2, this egg cell is released somewhere around the metaphase 2. It's not completed all the stages. And when cell is divided, the cell which is already dividing into two, and one cell which has become very smaller. So this one, the round shaped one, is a first cell, and the small round shaped one is a second cell. They have a very uneven division because one cell need a large amount of cytoplasm and stored energy food granules like a starch and maybe the lipid granules. The other cell become very smaller. It doesn't participate for the fertilization. And if I draw the nucleus, it is not exactly correct because during metaphase like things, this cell has chromosomes. And there's another thing of this cell is surrounded by a very special layer. This yellow color thick layer is called zona pellucida. And we can also see surrounded this zona pellucida, there are supporting cells which actually are visible during the maturation of the egg cells. And these cells still make another layer which is called corona radiata. Okay, we can see another structures inside this ovum. One of the vesicles, that mean small fluid filled sacs, which are located very close to the cell membrane. And these fluid filled small vesicles are called cortical granules. Cortical granules. Other than that, we can visible other structures like mitochondria, and also small lipid droplets which provide the energy for the cells. And I'm going to show some kind of a nucleus structure showing these chromosomes, because they are not completely finished the meiosis. Meiosis complete when there is a sperm cell only fertilize this egg cell. Otherwise, it doesn't complete the meiosis stage. Right, and the white one is the cell membrane. It is also called a white light membrane. It represents the cell membrane, right? Okay, this is the detailed structure of the ovum, and the small cell which is inside this, the big cell, is called first polar body. And this released egg cell is also called a secondary oocyte. Right, I think you should draw this secondary oocyte or ovum under the notes mammalian gametes. If not, I can provide this later, then you can draw this, right? If you have a time during this listening period, you can add this one. If you couldn't, I can give this one, then you can, looking at these structures, copy this down onto the notes, right? Okay, so we have learned the basic parts of these egg cells, and also a little bit background information which is important to understand. During the process of menstrual cycle, the starting days, these egg cells, several egg cells start to mature inside the ovary. Probably 20, close to 20 egg cells start to mature, and only one reach to the complete maturation process. After that, the 14th day or the middle of the menstrual cycle, this egg cell is released, and it is collected by these projections, and then movement of the cilia, it moves towards the middle of the oviduct. So the fertilization takes place at this place in the middle of the oviduct. If there is no fertilization, then this egg cell is disintegrated, and it has quite given lifespan, roughly around 24-48 hours, and after that it is disintegrating. So when the egg cell is released, and at the same time with this narrow window of 24 to 48 hours, if the sperm cell reach this area, then the fertilization takes place. So when the egg cell is released, it is still the stage of meiosis. So it doesn't complete the meiosis. As a result, we can see the first polar body. It's a division of these cells, and after fertilization, it make again small cells representing the completion of the meiosis. So these surrounding cell is also called theca cell. So they appear during the maturation process. So this theca cell create a thick cell layer during the maturation, and they're also responsible to produce hormone like estrogen and progesterone. Okay, so we now going to move to the next. We're going to see a sperm cell next. Okay, I save this slide, then I can send it to you. Right, the sperm cell is a male gametes, and the male gametes are produced inside the testes. So one of the difference of these female and male gametes, the male gametes continuously undergoing the process of mitosis and meiosis. That means if we take inside these tubes, so there are number of tubes, coil tubes, which provide large surface area inside the testes, and then these tubes has cells. So these cells can continuously divide by mitosis, and they can maintain the right number very close to the rim of the cell, the tube. So they are very special cells. They can divide continuously, and they make always constant number. While doing that, these cells promote some cells to the middle, undergo maturations. Once the cell enter into the middle, into the lumen of this tube, they are undergoing meiosis. So these cells are undergoing meiosis, and in the center of the cells, they completely they complete the meiosis process, and also they complete the maturation process, and finally enter into the last stage, that's the sperm cell. Again, we are not learning the production of the sperm called spermiogenesis, but instead we're going to learn the structure of the sperm cells. Comparing to the female egg cells, sperm cells are very small, but their number is large because each time they continuously male is producing a sperm cell by mitosis and meiosis, so their number is not fixed like a female. They have a large number of sperm cells. So at one release of the sperm cell, there are about 50 million sperm cell release. So then they are producing at large scale, and they are smaller. Okay, the sperm cell, we can find a nucleus, but the nucleus, as in female cell, it's in haploid, n number of chromosomes, half number of chromosomes, and this nucleus is surrounded by a special shaped cytoplasm. The cytoplasm is tapered like this, and it makes these projections and tip like a tiny tip to the front, and the middle again slightly specialized, and after that we can find a flagellum, and the front side again there is a fluid filled sac called acrosome. And here in the middle, we can find a collection of mitochondria. Right now, if you look this flagellum, it is important for the movement of the sperm cell. Unlike the egg cell, sperm is a motile cell, so it can move towards the egg cell. The movements are generating because of the special shape change of the flagellum. So in aqueous space, this flagellum can rotate and also move. Then it helps to move forward of the sperm cell. And mitochondria provide the energy for the movement of the sperm cell. And this front side acrosome has enzymes. These enzymes are called acrosine. We will see later on what is the function of the acrosome and the enzymes. And generally we can divide this cell into three pieces. This region is a head, and this one is called tail, and this one is called middle piece. Okay, if you copy this down to the notes, then we can move to the process of fertilization. Right, let's see the process of fertilization. As I mentioned you earlier, the ovary released the ovum during the middle of the menstrual cycle at 14 days, and this is collected by the projection of this oviduct or fallopian tube, and because of the movement of cilia and contraction of the fallopian tube, this egg cell is coming to the middle of the fallopian tube. Right, on the other hand, the sperm cells entering through the cervix, and the sperm cells coming from these directions toward the middle of the oviduct. So it should be here, the egg cell. Right, let's see how it really works. Do you see this video? Can you see the video?
Yes, sir.
All right, okay. See, here you can see this the cervix, and then it enters to the female reproductive systems, and it's roughly around 50 million sperm cells enter into the female reproductive tract. And then this one is actually no need to remember. Then the oviduct fallopian tube is again divided into several pieces like ampulla, fundus and isthmic. And here it comes to this region, the ampular isthmic junction. That means this is the middle of the oviduct. And you can see here the axil is coming from these directions. So as I mentioned, they have a secondary oocyte, and it is as the zona pellucida, and also it has this corona radiata layer. So now it first barrier is the corona radiata. That's a collection of the cells, and it passes through the corona radiata. Then it enters to the zona pellucida. This layer is the zona pellucida. Right now, what is going to happen? The acrosome, or the member of the cell membrane of the sperm cell, has receptors that can identify the receptors on the zona pellucida. That's identification. Once they identify this as the egg cell, otherwise, when the sperm cell is entered, it is so much of cells lining around this fallopian tube. They are different cells, and the sperm cell should find the correct cells. I mean, it's easy for us to tell this egg cell is the sperm cell, and sperm cell penetrate the egg cell. But how exactly the sperm cell find the egg cell is depend on the right receptors. That means sperm cell has a special protein on the cell membrane, and the matching protein expressed on the cell membrane of the cell membrane zona pellucida layer of the egg cell. So when they touch these receptors, it is the correct identification. Once it identifies, as I mentioned you before, these special enzymes on inside the head of this sperm cell called acrosine released. Acrosine can digest the zona pellucida. Here you see in this video, it is a digesting of the zona pellucida by acrosine. Once it digests, then it can push the head into the inside of the egg cell, right? So let's see the continuation. Yeah, you can see now the head of the sperm cell enters already to the egg cells. Right now it enters, but there is a small mistake of this video. Usually the whole sperm cell is not entering into the egg cell. Once once it break the zona pellucida, actually it push the nucleus into the egg cell. The whole sperm cell is not entering to this egg cell. Just only the nucleus enter into the egg cell. Right, once it enters, you can see the first polar body start to divide, and it's trying to complete the meiosis. Right now, next thing is, if you remember, I mentioned these cortical granules. So they are fluid-filled enzyme sacs lining close to the cell membrane of the egg cell. Now, what is the function of this cortical granule? When the egg cell break this zona pellucida, this activate these cortical granules. Then the cortical granule release enzymes. These enzymes responsible for thickening of the zona pellucida. So one, it can still close this the damaged area, When the sperm enter, it breaks this small piece of the zona pellucida, and it can replace that one, as well as they can make very harder the zona pellucida. Then the second sperm cell cannot enter. You see, it's closed now, and also it make very harder the rest of the layer, and it cannot break the zona pellucida now. See, yeah, now here it's correct. The two nucleus, pronucleus, it's called released by the first cell, and then these two nucleus going to fuse. That means the chromosomes numbers again pair up homologous chromosomes. Then it make the zygote. Yes, that is the end of the fertilization. And here it shows identical twins, how non-identical twins produced, how identical twin produce is always interesting question. So, any questions? You share. Okay, so if we want to still find the answer how the identical twin produce and how the non-identical twin produce, then it's like this. If this is the fallopian tube or oviduct, the fertilization take place at the middle of the oviduct, then that's a zygote. This is a fertilized egg cell, and the fertilized egg cell has again diploid number of chromosome. Then next, this cell undergoing mitosis. Now fertilization over, so mitosis takes place to grow the embryo, to form the embryo development. It's undergoing mitosis. So the first, what we can see, the first thing is then this zygote divide into two by mitosis. We know mitosis is production of two identical cells. Then if they produce two identical cells, usually the two identical cells we can show like inside like this, two cells. Then the two cells produce four cells produce eight cells produce 16 cells, 16 cells produce 30, like that. It's increasing the number. But a process which is very difficult to explain, and so far we haven't understand why this is happening. If the first stage of division, it become a two cell which is separated. These two cells separated due to a not so clear reasons. Then this cell start to develop separately, and this cell start to develop separately, because next stage is it's going to the wall of the uterus. So when during the process of journey to the uterine wall, they are coming to this stage with certain number of cells. So then they're going to embed onto this uterine wall separately. So they are already in the several cell stage. Now you see, uh, they now start to develop two embryos, and then this is explanation for the identical twins. One zygote divided into two, and these two cells are separated from each other, develop separately to become identical twins. Okay, we have to start the next session to find how a non-identical twins are produced. So this is the again ovum, and this is a fallopian tube. Right, how non-identical twins are produced. Now when we recall the meiosis has very two important stage, that is